in there at University of Washington. So she stayed on the West Coast. And then she switched back to the East Coast to start her first faculty position back at Harvard. And then apparently she decided to go, she likes cross-country trips. So at the beginning of COVID, she decided to move again, this time to UC Berkeley, where she currently is. She is a very accomplished scientist, and she's going to tell us a lot about her science coming up here. What really struck us is this combination of microbiology, but from sort of a more behavioral and integrative perspective. And so with that, I'm just going to go ahead and hand over the Zoom screen, and we'll let her take over.
Thank you. Let me just get this started. And Ignacio, can you see this? Okay. Well, good afternoon, everyone. And thank you so much for joining me today. And thank you to Dr. Ignacio Moore, the Fralin Life Science Institute at Virginia Tech, the Biological Sciences Diversity Committee, and the University and the Virginia Tech University community for this invitation and for this opportunity to speak with you. I would like to start today with one word: community. This simple word has many layers. As humans, we each come to the definition of this word with our own preconceptions of what it might mean and what it might look like now and into the future. For a lot of the work that we do in my group, scientifically research-based, as well as the work that we do as humans for outreach, we ask the question, what is a community? We ask questions about how it is formed, how it is maintained, how does it respond to external change? How do external pressures shape it? As we go through the science today, I want us to consider community from both the smallest perspective, such as that from a microbe, a bacterium, all the way up to the complexity of the human experience. And to be clear, I will speak from my own personal experiences as we talk through the science. But I'm fully aware, and I hope each of us are aware, that every single person has their own story and their own journey. We each have our own worries and our own pressures, and that is what makes us special. And that is something that we can bring to enhance the communities around us, how we respond to those pressures. So from the perspective of a microbe, once you have a single cell, or in the case of bacterium, a single organism, if you have more than one, so the second divides or interacts with something else, with another cell, it now becomes a group. And those and intergroup dynamics begin to emerge. However, group dynamics by themselves do not necessitate a community. So in preparation for this talk, I went back and I looked at what the definition of community is. And I like the ones put forward by the Oxford Languages. Their community is defined as a group of people living together in one place, especially one practicing common ownership. As we know, in an ecological or biological context, community is a group of interdependent organisms of different species growing or living together in a specified habitat. Given that this talk is part of the celebration of the legacy of Dr. Martin Luther King Jr., I wanted to also bring forward his idea of what community might be. Today, he brings us together through the extension of the idea of a beloved community, a phrase originally coined by the philosopher Josiah Royce. And here, the adjective beloved emphasizes the interdependence and love in this theorized community. And defined by the King Center in Atlanta, beloved community is used to describe a society of justice, peace, and harmony, which can be achieved through nonviolence. For humans, beloved can be a community in which everyone is cared for, absent of poverty, hunger, and hate. Yet building such a community and maintaining it depends on the actions of individual actors, each of us makes an active choice, either through action or inaction, often in the context of these group dynamics of pressures around us, some of which come from other living and creatures, and some of which come from non-living. And I think most of us in science would agree that our science, our research itself, doesn't sit alone. Science itself sits in the midst of all of these pressures and all of this human community. And recently I've been beginning to think about science as a multidimensional object, where it has several axes that are intersecting. Science sits at the intersection of community, family, friends, education. It allows us to explore the unknown, but our paths for exploration are often grounded in what we do know and what we have experienced. And so today, as I speak about my group's science, I want to also take a moment and talk about my community and experiences that I've had in both communities of the past and the ones that we're hopefully building here. As many of you might know, I was born in Jamaica and my family came here when I was a toddler. I grew up mainly in the Maryland and DC area. I still remember there being farmland not so far of a drive away from my childhood home. But like many children of the diaspora, I spent summers with my grandparents in Jamaica. During the weekdays, I was sent to school in the summer, which was not always fun. And then on the weekends, I was able to play on their farm. You can see evidence of some of my experiences in Jamaica, where every once in a while I forget to remove my U's from words, or my children tease me a little bit about my calling the Z-axis Z, not infrequently, or my love of pears, or as they call them here in California, avocados. However, when I look at my journey in science, it first started really when I was in high school, where I was fortunate enough to benefit from programs through the National Science Foundation and the Department of Defense to introduce high school students to bench research. And after 2 summers of working in a Department of Defense laboratory under the supervision of Mr. Wayne Kell, I realized that science, research science, where you're pushing the boundaries of the unknown, was something that you could make a career out of, and something that I personally enjoyed doing. Looking through my journey, along the line, several individuals helped or guided my decision-making. So in college, I ended up choosing my research lab based on the advice of a freshman advisor who encouraged me to speak to the young Colombian professor about his research. And that's when I fell in love with collective behaviors, studying biofilms in Sumans ruginosa. I ended up going to graduate school at Stanford on the advice of Dr. Lucy Shapiro, who convinced me that that program at that time was a place where they invested heavily in individual graduate students. I worked with Dr. Pete Greenberg at the University of Washington in Seattle for my postdoc, studying how organisms talk to each other. based again on the advice of Dr. Shapiro, since she knew that I was really interested in those social interactions. And then Pete influenced me probably the most in forming my lab group as it is now, because he was trained with Woody Hastings when he was a postdoctoral fellow, when Pete was a postdoctoral fellow at Harvard. And Pete often comes back to this quote: "The great thing about Woody Hastings during my era at the biolabs at Harvard was that he never forgot that science is supposed to be fun. Ambition was kept in its place." And that is really the way in which I approach my science. What you see here is the community that the students and postdoctoral fellows, undergraduate through technicians and postdoctoral fellows, have built over the years. We're a group where people are recruited and accepted for who they are by not the content of necessarily what they say, but what their actual actions are, what they do for themselves and for each other. We are a group that are people who have been in America for several generations, and this is their first time. Those that stay, those that are young, those that have chosen to stay in science, and those that haven't. So the work I'm going to talk about today was primarily done by Dr. Untitled Chapter. So she's here in the middle. She is the young woman to the left in this image. She was born here, but her family was originally from India. And then the other work I'll tell you about is from Dr. Murray Chipping, who is the gentleman down here on the bottom right. And he's originally from Scotland and now has made, from a small town in Scotland actually, and has now made the United States his home. Before going into the science itself, I just wanted to briefly talk briefly about one aspect of the science that I encourage in my group, which is that we really believe in reaching out and contributing beyond the research that we do. And so the majority of students and postdoctoral fellows in my group have done community outreach. I give them time and sometimes resources to help do that. For myself, I try and support those students and colleagues, not just in my department, but in the broader community as well. I think it's really important as young scientists to remember that as much as we want to focus on our science, we have expertise and energy to share with people beyond ourselves. And of course, that can ebb and flow at different periods in our life. But I encourage you that to understand that, I encourage you to explore the idea that sharing science beyond just what we do on our benches can actually bring fulfillment back to ourselves, as well as that for our community. And then finally, before I go any further, I want to take a brief moment to thank and acknowledge that Berkeley, the University of California, Berkeley itself, sits on the ancestral lands of the Ohlone people. And that the Ohlone people are not just in our history, but they're living here in and among us and contributing to our community at large. I want to also apologize. As I said in the meeting with the committee ahead of time, they're doing construction in the building right behind me, and today they're doing the sewer laterals. And so if there's a little bit of noise in the background, that's what that is. So launching a little bit into the science itself, I started off by talking about communities. And as I said before, I'm really interested in community dynamics. And more importantly to me, is how does an individual go from being a single to being a collection of individuals who work together or can work together to achieve a greater end. And that is sometimes referred to as collectivity. We know in the bacterial world that we see community behaviors or collective behaviors in different environmental habitats. For example, we can see it in the soil, so in the roots of plants, you can see biofilms that form and provide nutrients for the plant themselves. We see it in the organisms that live on and through our bodies, whether or not you're an animal or a human. For example, right now in the news, you can hear a lot about the commensal bacteria that live in our guts. Those are the bacteria that help degrade some of the foods that we eat. Many of them are capable of collective behaviors. And we also know that collectivity and community behaviors are important for virulence. And so, for example, in biofilms where bacteria can form these incrustations either in your eye or in other places that cause disease, they can talk to each other, and through that communication, activate pathways necessary for virulence. I study an organism that is found in your gut called Proteus mirabilis. And when it's in your gut, it does not cause disease. It's thought to be either avirulent or commensal. However, when it's able to enter into its collective form, it's able to move as a group. It's thought to move as a group from your gut to your bladder, where there it causes disease and becomes virulent. And its movement to virulence is very much connected to its collectivity. The genes that are turned on to cause disease are controlled by the genes that lead to this cooperative migration. Now, Proteus mirabilis is called this after it's a miraculous changing, shape-shifting, like the Greek god Proteus. On this image, on the there are 4 microscope images that you see. On the top left is a phase image, so the cells are dark bodies on a gray background. That's the nutrient agar they move on. To the right of that is an image taken in the red fluorescent protein channel. These are cells expressing just the red fluorescent protein. On the bottom left panel is from the green fluorescent protein or GFP channel. Again, these are cells that are expressing just GFP. And on the bottom right is an overlay where there's the cells that are expressing GFP are in green, cells expressing RFP in red, and everything else is, every other cell is dark. I bring this slide to show you 2 different things. First of all, is that the reason Proteus is called this is that the cells, instead of being what we think of as kind of standard bacterial short cells, they can grow up to 40-fold in one life cycle and become long snakes. And these long snakes are best visualized in that RFP or GFP channels. And you see that these snakes are flexible. They're not just long, they actually twist and burrow. They can fold back on each other and form a little almost sandwich. They can be S-shaped or hook-shaped. All of those cells are just a single body, one cell, but many different shapes. These cells in this developmental cycle are just covered from head to toe in hair-like structures called flagella, and these allow them to move and crawl across the surface. Proteus, when it's in this infectious state, this is called the swarming state. They're found on top of the surface itself, and they move on the surface between the air and the surface. The other thing is that these cells, when they're moving, they're cuddled against one another. They're not sitting out there like individual butterflies flying off on their own. They're colliding and sliding against one another. And in fact, their collective this collective motility requires cell-to-cell contact. And that's most best visualized here. Again, this is a phase microscope image where the cells are dark and they're moving on top of the gray background. And the slide is sped up about 10x, and there's a black screen every time the movie repeats. And I hope you can see that these individual cells, they're moving, gliding, touching one another. And this motion on the micron scale, where they can go several tens of microns at a time, forward or back, allows them as a group to occupy centimeter scale distances in the course of a day at human body temperatures. Again, as a collection of cells simply migrating across one another, these organisms are able, these cells that are only 10 to 100 microns long, are able to move up to a full 5 centimeters in under 24 hours at the temperature inside of our bodies. That's visualized here on this movie taken by a former graduate student, Dr. Jacob Osterman. On the plate on the left, we've inoculated 2 colonies of Proteus. This colony of Proteus was taken from, these 2 colonies were taken from the same human. And indeed, from the best data that we have, it seems that each of us have our own individual genotype or strain of Proteus. So that inside of me is different than that inside of any one of you. And these Proteus, sorry, this is mislabeled. I'm sorry, there was a slight mislabeling on that one. And these Proteus cells, I'm sorry, I'm going to stop for 1 second because there's a mislabel, and I really dislike when that happens. I got this slide from a student, and I didn't double check it. I apologize for that. And so on the image on the left, I should say, the bottom one is actually from any one of us individuals, and the one on the right in orange is from a different individual. The side on the image on the right actually is the one where both populations are taken from the same individual. Again, on the right with the two blue markers are the populations taken from a single individual. When those populations migrate out, they form this bullseye pattern. And this bullseye pattern is due to an oscillation from being these long cells that are rapidly moving that you saw in those movies, and periods of when they stop and divide into short cells that don't move. And that gives a very stereotypical concentric pattern to the colonies. When colonies that are swarming that meet from populations that are from two separate individuals, as displayed on the left, a boundary is formed. And this boundary is visible to naked eye. We now know from work from my group and other groups that this boundary requires that the cells actually touch one another, and that it formed after several hours after cell contact. And so I'm going to play this movie one more time so that you can see it again. And this is taken over several days of room temperature. If you focus on the image on the left, you can see that the cells actually interact with each other quite a lot before a boundary actually is visible. And that boundary is absent from the population where the cells are from the same population. We now know that the boundary is due to an exchange of proteins directly from the inside of one cell to another cell, and that these cells are curled up to one another. And so in this image here, the yellow cells are from the orange population and the black cells are from the blue populations. And this is taken from what we call the protoboundary before you even see a boundary form. So for the remainder of this talk, I'm going to answer two questions. How do cells distinguish self from non-self? And how does this information impact population behaviors? We know from the work in my group and others that this is due to communication. There are two types, and this communication can happen in two different ways. There's a lethal mechanism in which cells actually take a sword called the type 6 secretion system. They add a poison pill to it. And then as they're sliding against each other, and only when they're sliding against each other, they stab each other with a sword and drop off that toxic payload. If you are a sibling, you are making an antidote that allows you to degrade the toxin. In the case of Proteus, this strain of Proteus, that toxin is a DNA targeting, DNA degrading enzyme, and the antitoxin binds it and leads to its degradation. If you are not a sibling, well, you die because you have no DNA. We're not going to talk about that today because, you know, in the words of George Washington, taken and interpreted by Lin-Manuel Miranda, dying is easy, living is harder. And so instead, we're going to focus on the work in my lab, looking at advertisement or the non-lethal, non-killing ways in which bacteria, specifically Proteus mirabilis, is able to distinguish self from non-self. Much of the work that I'll talk about today has been previously published, and so I'll give you an overview, and then we'll dive into some of the newer data that I hope will be of interest, and I'm happy to answer questions about anything I present at the end of the talk, as well as where we're going next with this. So we now know that in the strain of Proteus mirabilis I study, as well as in over 90% of the strains of Proteus that we've examined, and we've examined almost 200 of them in nature, that there are two non-lethal factors that encode identity information. They're found next to each other in the genome, and they're part of the same translated area or oplon. One of them is called IDSD, and that is depicted here on the left. It is a 114 kilodalton protein that has two predicted transmembrane domains labeled here TM1 and TM2. For the remainder of the talk, I'll refer to this as the message. IDSD has over 96% conservation across these different strains that we've looked at, except in the region flanking that second transmembrane domain, where it goes down to less than 70% amino acid conservation in pairwise comparisons. This protein is exported via the type 6 secretion system and delivered directly into the interior of a neighboring cell, again on that sword. In the neighboring sword, there's a receptor or a glove mitt waiting. And that receptor is called IDSE, depicted here on the right. IDSE is a much smaller protein, it's only about 37 kilodaltons. It has two predicted transmembrane domains. And like IDSD, IDSE is over 96% conserved over the vast majority of the protein in pairwise comparisons. But when you look just at the region flanking transmembrane 2, especially in the paraplasmic loop and the transmembrane domain itself, we get less than 30% amino acid conservation. In each strain of Proteus that we've looked at, isolated from humans and animals and all sorts of things, they have one gene that encodes IDSD, but several genes that encode IDSE. However, ADC. For simplicity, I will depict IDSD as a rectangle for remainder of the top, and that variable region, the area of lower amino acid conservation, will be a darker band. Likewise for IDSE, which I'll refer to as the decoder, it'll be depicted as an oval with the variable region band, again, being a darker color. To summarize what we've shown directly that only in Swimmer cells do we see, so those long cells that are migrating and cuddled against each other, those are this infectious stage, not the non-infectious stage in your gut. Only in these Swimmer cells do we see the transfer of IDSD outside of the cell via type 6 mechanism, where it gets stabbed into the neighboring cell and it's able to bind the receptor IDSE. If every cell in the population has both the blue decoder and the blue message, you get a migration where the population is able to cover an entire 10 centimeter diameter plate in 24 hours at 37 degrees Celsius, the body temperature. In other words, the entire population is able to move outwards and occupy that 10 centimeter diameter space. Through biochemical work, we have shown that a blue message will bind a blue decoder, and an orange message will bind an orange decoder. However, there is specificity in this binding, which you can see on the bottom part of the slide. The orange message will not bind a blue decoder biochemically, and the blue message will not bind an orange decoder. If you take the blue decoder and simply swap out the variable region and make it that of a different sequence, for example, that of the amino acids from the orange decoder, they don't bind. But if you now swap out the messages variable region and make it match that of from the same string background as the variable region of the decoder, they'll bind. And so the entire 4.5 megabase pair genome can be simplified down in Proteus, at least, to having identity determined by the amino acid sequence in the two variable regions, that of the decoder and that of the message. So what does this mean for population interactions? Several years ago, one of my graduate students, Christina Fox, said, well, we have the genetics now in Proteus. Let's ask the question directly. If you have a population where the entire genome is the same, except for just the 80 or so amino acids that constitute the variable region of the decoder, what does that do to the community? So what this means is that every time a cell is moving out, it's getting jabbed by its neighbor, and it receives a message that it can't interpret. And what we found was that there was an 80% reduction in collective migration. We didn't see an increase in loss of life. We didn't see a differentiation between becoming long and short, so the cycle of the development. But we saw that the population as a whole only went out about a centimeter, maximum 2 centimeters, rather than covering the full 10 centimeter diameter plate. If we remove the sword, that type 6 secretion system in this genetic background, we found that you were now able to rescue migration. And the cells became, they were able to expand out fully once more. So it was receiving the message that they didn't understand that allowed the population, that restricted the population's migration. But the second that they're not receiving this message, they're fine. And you get collectivity. That's also the case if you just remove the whole system, if you have no identity. Yet we can only we found less than 1% of Proteus populations in the world that at least from the sequence populations available through either NCBI, our own private databases, and others, less than 1% of them don't have identity proteins. So for this, these proteins are found predominantly across the populations, but they don't need them. And if they receive the wrong one, it screws them up. So Achala asked the question, how can cells escape this identity-based regulation of swarm expansion? How, if you are stuck in a situation, always receiving a message you don't understand, how do you escape? Can you return to the collective behaviors? And the answer is yes. You can return in many different ways, one of which is, of course, to turn off the type 6 system, one of which is to get rid of the proteins, which I've talked about already, but there's a third way. And that's the one I'll tell you about, dive into that data right now. So what Achala did is that she used an unbiased evolution experiment where she was looking for the emergence of suppressor strains, starting with with a population where every single cell is receiving a message it can't understand. She allowed this population to sit on her bench at room temperature, parafilmed, and grow. And after a week, which happened to be, I think, Thanksgiving or Christmas, she came back. And what she found was that out of that population emerged these spurs, indicated by the arrows, where you get migration. And you get this collective swarming outwards, again, showing the concentric pattern, but you can tell that they're different from one another because you get different sizes and shapes and they do it at different speeds. She did this independently several different times with different plates, and taking a single suppressor from all of these different independent evolution experiments over and then mapping them using whole genome sequencing over and over again, she found mutations in a single gene called SDAC depicted below. In black are all mutations that she saw that were deleterious, to stop the gene from working. And in pink were the 2 mutations she found which allowed for full-length protein to form, but still led to a non-functional protein. She confirmed that in fact SDAC is the causative gene for this by doing a deletion complementation. So on this graph on the y-axis is the swarm radius, so that's the distance taken from where we inoculated the plates to the end of the swarm migration, as depicted here on the bottom plates. In each column is the samples that she did. So in the first column is the wild type population, BB2000. Every single dot is a single experiment. The black bar across in the graph itself is the average. What she found was that the wild type was able to expand almost 25 millimeters on average. With the non-self populations, in this case, we actually removed the decoder fully to really enhance the phenotype. And we see the lack of swarming, as we talked about before. In this non-self background called NS, she then made a clean deletion of this SDAC gene, and she saw that that was able to complement back to wild types. The cells were able to expand out with that collective migration. And then if she reintroduces via plasmid under the control for artificial promoter or under control of the nature promoter, the SDAC gene, she's able to again see that restricted collective migration. SDAC in E. coli was not at all studied in Proteus, and so we had to turn to the E. coli literature. And what we know from E. coli is that SDAC is the Syrian export uptake transporter. So in these bacteria, E. coli and Proteus, these cells actually have 2 membranes. They have an outer membrane. There's a space. They have an outer membrane. There's the periplasm space, inner membrane, and then the inside, the cytoplasm. To get L-serine from outside your cell to inside the cell where the cells need to be able to eat it, you have, excuse me, you have to have a dedicated transporter. In this case, this is SAC, and it sits in the inner membrane. And what that does, what SAC does, is that along with the proton, it's able to pull L-serine and pull it into the cell. In E. coli, one way you can demonstrate that it's a serine transporter is that you can get rid of the serine deaminases, which is called SAA and SAB. So these are the genes inside of the cell, the proteins inside of the cell that are necessary for chewing up and eating that serine molecule. If you get rid of them in E. coli, it basically causes E. coli to be really stuffed. The serine SAC, it doesn't respond to the fact that you have too much serine on the inside, so it keeps them stuffing itself. And so I like to think of it like cotton candy, kids at a fair with cotton candy. It just keeps on stuffing itself with that cotton candy, and the cells go under a lot of stress from too much serine, and you can measure it. And so we directly measured that in Proteus using strains that Achala made. She looked at a strain that was wild type. She looked at a strain in which she's deleted both SAA and SAB, which is called the high serine. And then that high serine strain, she got rid of SAC. And we directly measured the amount of L-serine within the cells using mass spectrometry. And what we found was that if you look at the amount of serine within any given population, a swarming population of the wild type, this is the basal level. We see almost 1,000-fold increase of internal serine when you get rid of those serine deaminases, so you don't break it down. But if you get rid of SAC in that high serine background, it relieves the pressure. And the cells return to more like wild type level of serine. So SAC is not just a serine uptake transporter in E. coli, it's also doing that in Proteus. She then went on to show that, so I've shown you so far that SAC is needed for escaping control in the non-self cells, and that's responsible for L-serine transport. She demonstrated this also with growth curves in minimal media, plus and minus excess serine. She went on to then further show that by deleting the other serine transporters in Proteus, you did not actually affect serine levels during swarming. And indeed, SAC, when the cells are migrating across, the game in town for eating serine is SAC. It is the primary transporter. And it acts like this uncapped hose where it's just sucking in any external L-serine that gets in the cell, it's just sucking it in and sticking it inside. And that this too much serine, it can cause cell envelope stress, causing cells to stop swarming. And that's through disrupting cell wall synthesis and some other aspects there. But that is not actually the cause of the escape, the non-self escape. The serine transport itself, the utilization of serine itself, is not why the cells are escaping. And indeed, it's not even that it's causing the proteins, the identity proteins themselves, to be produced differently. These serines affected these cells where we disrupted aspects of the serine pathway produce the same amount of self-recognition protein as the wild type, and they export it similarly. So how does SDAC actually prevent this recognition-based control? Well, SDAC is a multi-domain protein. It sits multi-domain protein that sits in the inner membrane. It has it has several passes of transmembrane domains, and a model of it using ITASR is depicted here on the left. This SDAC is a well-conserved member of the lutefold family, and so the model developed by ITASR was predicted to be very high. And how it sits in the membrane is that it has loops that face out into the periplasm and loops that fit on the inside. From work from Rochelle Godet as well as others on lutefold proteins, we know that wild type lutefold proteins transporters act like the following. In an open conformation, their loops are open and wide in the periplasm. That allows for serine and a proton to come and sit in the pocket. Once serine and a proton are there, the loops will close and basically do a jumping jack. And that will open up the loops that are in the cytoplasm, allowing for serine and a proton to transfer into the cytoplasm. Thankfully, all of this work was done in other systems. And so then Michelle was able to use the biochemical and structural work and map that onto our predicted protein. And she made some very, well, we made several mutations, but the ones I'll show you about today, we made structural, we made functional changes to the protein in order to determine how it's acting in self-recognition. And so we, to summarize the data, again, this is now published work, which you can see in the Journal of Bacteriology. She made mutations that would change the transport status and lock them in either open or closed or flexible formats. The serine transport was measured using growth curves in minimal media and serine. So we have normal serine transport for the black wild type protein. We can make a mutation in this valine 222W to tryptophan that locks the cells, biases them in an open conformation. And when we do that, you don't get any serine transport. We can reduce the amount of serine transport that's happening, and especially if you have a live external amino acids, drive it down to almost nothing by widening up that pocket where the serine sits. And so basically not just serine, other things can sit in there, and so it gets outcompeted. And then we can lock it in a closed format or bias it in that format by making these glycine mutations below. Ironically, these glycine mutations that you see right here, or maybe serendipitously, these are the same mutations that she isolated out of her unbiased evolution. In other words, nature gave us these mutations in addition to the work based on other colleagues. We then introduced each one of these independently in the non-self background in the absence of the wild type SJC to ask how these mutant proteins are able to contribute to that collective migration. And what we found was as follows. On the y-axis, again, the swarm radius. On the x-axis is each population. I want to remind you again that this is in a background of cells where every single cell is receiving a message it can't understand, and we've removed the native SJC protein. And that is how we found, and we see that they have normal wild type like collective migration. If we add back the wild type SJC, as you can see here in the second column, that reduces the ability of the population to expand. So they're constricted. In either the green or blue mutations, in which the population is able of SJC proteins are able to sample or be biased into an open conformation, they look like the wild type in terms of swarm migration. And only the cells in which SJC is in a closed conformation are able to look like the null mutation and rescue collective migration. In other words, if this population is able to, if SJC is ever open, that is sufficient for the cells to receive a message, to interpret the message and not migrate. We wanted to know if it's something special about SJC itself, this protein in Proteus, or is this just any serine transporter? And so we used the protein from SJC, the wild type SJC from E. coli, which, and then we, as a protein that's like SJC, and we used YHAO, which is from Proteus mirabilis, another serine transporter that's a lutefold protein. On the left here is just showing you that they have many of the same folds, and on the right is the alignment of their predicted models. Again, they have generally the same shape. Of course, the amino acids, there are differences between them. The prediction, of course, is that if it's from E. coli, they are really far away in the evolutionary tree. It shouldn't work. And in fact, however, again, when we introduced these proteins from SJs from E. coli, or the YHAO protein from Proteus, we were a little surprised by the results. On the left is again the graph. On the y-axis is the swarm radius, and on the x-axis is each of the populations. We introduced each of these proteins into that non-self background where they're receiving a message they don't understand, but they've removed the SDAC, the wild type SDAC gene. In those situations, we found that now when you, and so again, this is wild type SDAC from Proteus. We found surprisingly that the E. coli SDAC acts just like the wild type SDAC. In other words, the E. coli protein is able to act in this recognition pathway. However, this other serine transporter lutein-fold protein, YHAO, doesn't. And so in summary, what that suggests is that this open conformation of SDAC might be acting as a landing pad for the message as it enters into the cell. We think that our model is that what it's doing is that this open conformation, which is residues that are actually probably conserved between E. coli and Proteus, are allowing for the message to get close enough to the membrane and allows for then the message to be integrated into the membrane and bind and interact with its decoder protein. However, of note for those of you who are interested in kind of metabolism ecology, it's interesting that this message is targeting the serine transporter, the SDAC, because we know that serine metabolism is an incredibly important part of virulence for Proteus when it's inside of the bladder. And moreover, we know that SDAC, this is the dominant transporter that's present when the cells are actually doing collective migration. So in other words, we've intersected this messaging system is pulling together not only collectivity, but it's also directly sitting itself on top of metabolism and virulence, both of which are necessary and important during periods of collectivity. But this doesn't really tell us what's happening in the non-self situation. Why do the cells stay constrained? Why don't we see collective migration? We know we can escape it by getting rid of your serine transport, even though that has other problems. But why? And so in the last few minutes here, I want to touch on the work of a former postdoctoral fellow in the lab, Murray Tipping. And he asked the question of how does it regulate swarm expansion? How does identity add to community formation? And to summarize his work, because it's published at this point, he found that when you look at the RNA-seq of these populations, that populations that are receiving, delivering and receiving messages they can understand, they turn on the genes necessary for efficient migration and for the production of these virulence factors. However, in populations that are continuously receiving messages, which they can't understand, those cells, and only under consistent pressure of receiving these information, increase the production of an alarm molecule called PPGPP. And this leads to increased antibiotic persistence and decreased metabolism. And I just want to demonstrate briefly what I mean by increased antibiotic persistence. If we harvest cells from swarms of everything self, everything having no identity or having non-self, and measure survival over time, what we found was as following. On the y-axis is colony forming units per mil, in other words, survival. And on the x-axis is time after we've hit them with lethal doses of antibiotics. In the black box, and in the x's, in the black box are cells where they're receiving messages you can understand and interpreting them. And the x's are, do you have no identity? And these are all populations that swarm that full 10 centimeter diameter plate. You see a rapid die-off in the presence of antibiotics. However, in a population, when cells are receiving messages that they don't understand, so every single cell is receiving a message it doesn't understand, this non-self population is able to resist death by antibiotics by about 50-fold. If we delete SDAC in that population, it reduces it back to the wild type. And so receiving this message is actually providing this uninterpretable message is driving the cells into this protective state. And indeed, there's a prediction that comes from that, which is that if every cell that's receiving a message that it can't understand is led to being in a more antibiotic resistant, protective kind of biofilm-like state, then if you mix them together with the wild type population, those cells that are receiving messages that you don't belong, they should be left out. They shouldn't be able to get access to the outer edges of the swarm because they're not turning on the genes necessary for efficient migration. And indeed, we can test this by mixing together populations that are 50% giving messages that they can receive and 50% no identity. The population that's receiving the message that they can understand can be in red, and a population that has no identity and so is constantly non-self is in green. We actually only have about 10% of the cells labeled, so we can do single cell analysis with them. We measure the population, what these populations look like over the course of the swarm. I'm showing a complete form plate at the bottom to give you an orientation of where we're looking at. So if you start at the very center and you look at that first swarming, when these cells are in 50-50 population, you can see many of both the cells that are self and the cells that are non-self. But by the second swarm front, what you find is no longer are there cells, as many cells that are receiving the non-self message. And by the third swarm front, those non-self cells are hard to find. If you calculate them directly by a ratio, and the ratio here is on the y-axis of non-self to self, well, and this is on the x-axis is time and space. So in gray are periods of migration, and white are periods where the cells are stopped. And on the far right is the final geomapping of the self and non-self population. What we find is that over time, those non-self cells are left behind. They're excluded from the leading edge. And this is not the case if the cells are all self. Murray went on to show that what's happening in those non-self cells is that during periods of migration, they're migrating just as well as the cells around them. But when the cells have to stop and divide, at that point, that's when they turn on these genes indicating stress, indicating that they've received messages they don't understand. And rather than continuing on and re-entering into the developmental cycle, these cells that are different, these cells that are receiving that non-self message, get kicked out into a different developmental stage. They're left into a developmental stage that says, stop, protect yourself. You're around something different. Why does this matter? Well, in microbiology, we know that there are many ways that bacteria use to recognize each other. There are things like form sensing, which are a diffuse signal where it's basically shouting in a crowd, and cells that are able to interpret that message are able to access the collective grouping. We know both in bacteria and eukaryotes, such as Dictyostelium, that there's signals that are on your cell surface that allow cells to transiently adhere to one another. And if you are kin and these recognition signals are the same, you adhere and are able to engage in either migration or, in the case of plants, nutrient sharing. For Pervious, this is an example of a non-adherent signal, one where a signal is dropped off during periods of migration, but only interpreted during periods where you're dividing. We think that Proteus does this because, unlike those other organisms, Proteus's time of collectivity is on a surface. Proteus, when they move and need to do group behaviors for migration, is when they're moving rapidly together. And in order to move rapidly, you need your neighbors. On this microscope image you see here that was taken by a former student, Martha Rivera, the gray is the background. Each individual cell is embossed. Green, about 10% of the cells are expressing green fluorescent protein, so they're green. Cells that are dividing and not moving are on the bottom left, and cells that are actively moving and migrating are on the right. This migration requires each other, but it's also on a surface where small molecules can diffuse away rapidly. So for this organism, how it's determined to build this community is that by dropping off packets of information, whether you want it or not, physically jamming the message into a neighboring cell as they move, leaving breadcrumbs, internal breadcrumbs. These breadcrumbs tie into their nutrient system, and only if you're a sibling do those breadcrumbs encourage, allow re-entry into this collective elongation. And so we've termed the term micro crowdsourcing as a way of indicating how swarming bacteria are able to integrate signals from inside themselves to build local migration communities as well as macro scale. And so I want to end by spending just one more minute speaking about those who support us. As I said before, the work that I do here is not my work alone. It's built from a community of scientists, both within my group, as well as our conversations with our colleagues in my department former department at molecular and cellular biology at Harvard, where all this work is done, as well as colleagues across the world. It's supported by agencies and institutes and private foundations. It's supported by being able to speak with community members at large, those who are not scientists at all. But I also especially want to thank those who support us who might not always get the recognition. And at this point, I want to highlight the work of Mrs. Coretta Scott King, and to bring this talk in honor of Dr. Martin Luther King Jr. to a close. She was his supporter and partner in life and work. Mrs. King lived at the intersection of her femininity and her race, and she elevated above in a time when few others did. Like Shirley Chisholm, Barbara Lee, Vera Rubin, and many others, Mrs. King surpassed the prejudices of others, and she made a table for herself and those around her, rather than waiting for a chair to be pulled to the tables that were present or to be called to action. She was the first woman to speak at a class day at Harvard. She was a renowned humanitarian. She was a personal witness to notable moments for people-driven communities, such as the democratic election of Nelson Mandela after the fall of apartheid. I think it's notable that above Dr. and Mrs. King's script, inscribed are the words, quote, "And now abide faith, hope, love. These three. The greatest of these is love." And I encourage all of us to move forward with a love for humanity, a love to understand that each of us brings strengths and each of us have our own weaknesses. And so I will leave you today with the words of Archbishop Desmond Tutu, who frequently espoused that each of us should do our little bit of good where we are. It's those little bits of good put together that overwhelm the world. And for me, this means that each of us individually make a choice each day to build and to maintain our communities. Each of us can help ourselves and our science thrive. I look forward to hearing more about your continuing successes as we move forward in your lives. Thank you.
All right, so thank you for that wonderful talk. We'll now start the Q&A session. So for the audience members, if you have any questions, feel free to type them into the Q&A little option down at the bottom, and then I'll be able to read them out and deliver them to our speaker. So we have about 2 or 3 up here right now. So I'll go ahead and ask you the first one. Is there an active mechanism to make the hypervariable regions more variable, or is it just based on average mutation rate?
So we suspect, but we do not have evidence that there is something strange going on. So there's a higher mutation rate at that region. So if you look at the genome itself, there's a standard mutation rate, and this is above that. But then even within these genes themselves, so how the IDS region is, the locus is oriented, is that you have two genes necessary for transport, a gene necessary for protein folding of the message. Then you have the message, you have the decoder, and then you have another gene necessary for transport. Every gene has over 96% conservation, even down to nucleotide level for the transport genes. The chaperone has a little less conservation, but it's fairly highly conserved. And then even the message and decoder are highly conserved on the nucleotide level, except for the variable regions. And so there's just this span of about, a couple hundred nucleotides that's conserved, variable conserved, conserved, variable conserved, where the mutations are just out of the world. So we thought for a while, well, maybe this is happening through, essentially invasion from foreign IDSE or IDSGs. And we don't really see evidence for that. We do see evidence that they can acquire additional IDSEs from the environment. However, their additional decoders from the environment onto their genome, but those decoders don't bind the message that they make. And so we don't think it's through horizontal transfer that you get those variable regions are as they are. Now, what is that active mechanism? We don't know. And clearly we'd love to find it.
So here's another one, and I was also wondering about this, is what happens to migration when a self and non-self strain both with active signaling start from the same spot? And what does that population structure look like?
Right, so that is, actually, I'm gonna stop sharing for 1 second, and that's a great question. And what I'm going to do is actually pull up and show you a video of it. And I should have put this as a hidden slide because people ask, but then I forget. So give me 1 second here. I apologize. Let me get, I apologize for the delay. So the population structure becomes such that the non-self population gets constrained to the center, given everything else being equal. What we have found, of course, though, is that there are more than one factor that's involved in recognition. And so, of course, if you have multiple lethal factors, what can happen over time is that the cells will be killed off. And it all depends on the emphasis of on how well the killing action is. However, if you have, essentially, if you get rid of the killing action, so the lethal toxins, you find that the non-self population stays in the center. And I apologize, I wasn't able to pull about, I'll keep going with questions, and if I can rapidly find the movie I wanted to show you, I will, no, there it is, okay. And so this is work that was done. This is all preliminary data, so please take this with a grain of salt. This is work done in collaboration with Michael Boehm at Harvard Medical School. In this situation, we take a self population that's going to be all in red. And so these have a message and a decoder that they can talk to each other. So all the red cells can talk to each other and exchange private messages. And then the blue population is that non-self population where they don't have the decoder, so they can't interpret the message. And for this blue population, we have put a reporter gene on the chromosome that layers up anytime they receive a message that they don't understand. So they're getting that stressed out message, and they're engaging in that alternative lifestyle. We mix them at a 1-to-1 ratio, and then we measure over time. On the top is just the light image, and on the bottom is the 2-color overlay. So again, red, those are all the cells that are self, and in blue are the cells that are non-self, but are also stressing out about it. And what you can see over time is that, very early on, you get a large population that is non-self and expressing the message. But by the time you get to the end of this migrating colony, you get the presence of, you get fewer and fewer of the non-self population, but you still get pockets of them. So you get what you get sectoring essentially of this effect. And we're now going down and looking at it on the single cell level to try and understand what's happening with the sectors. And it's a little surprising because we don't actually see sectoring when we label the cells with any fluorophore. Usually get mixing because the cells are swarming around. And so the fact that you see sectoring under this situation suggests that something else, there's some underlying factor, and we don't know what that is. That was a long answer. I'm sorry, Dr. Moore, but I wanted to share this image.
Long, but very cool, and I do appreciate the video. Looks awesome. The next question that I have is, are you actively working to prove that the SDAC protein, the SDAC protein, is interacting with the message?
We are, and so we have some preliminary data with biochemistry that the SDAC protein is able to bind directly to the message. Ironically, though, we cannot actually see the message. So we can see, and so to do this, we do things called pull downs. So there are immunoprecipitations where we basically take our SDAC protein that's has a tag on it, and we use it like the bait on a fishing hook, and we throw it into a milieu pond, and we're able to pull out the message. However, if we reverse that experiment and we do it with the message, we don't pull out SDAC. And we think that it's because it's probably a transient interaction where there are other proteins that are involved in it. And so right now, one of my students is gearing up to do a whole series of bacterial hybrids and similar to try and show a direct interaction in vivo, which would, of course, that's what we aim for. Sorry, I'm laughing at the next question.
Yeah. The next question that I'm going to ask you is, why are there multiple genes for the decoder?
Yeah, that's a great question. We don't know. I'm sorry, that's an unsatisfying answer. That is a great question. And honestly, we don't know. And I think the thing that's striking to me is why wouldn't you just have multiple copies of the message? We know that for the lethal factors, we can have many copies of lethal factors, right? Cells, they'll happily have many things will kill other cells. But for the non-lethal factor, there's the one. So I think it's not just a question of why are there multiple alleles for the decoder, but perhaps why is there only maintained one allele for the message? The other thing is that we thought for a long time that maybe the multiple of the decoders allowed cells to basically go incognito, right? So maybe you can get away by giving a message, but then you can sneak attack and hide out. We don't see expression of the other decoders when you actually measure by RNA-seq, and so we don't. Unfortunately, that model did not suffice. So I don't. It's an open question. I just don't know the answer. I'm sorry.
Completely understandable. So the next question that we have is, I wonder to what extent you see parallels between bacterial population structures and human population structures and how individuals interact.
Yeah, so thank you for that question. And I will preface my answer as following. The beauty of humans and many eukaryotes and animals is that we have consciousness. We have brains. We develop over time. And as far as we know, bacteria do not have consciousness. As far as we know, they don't have brains. And so what we are seeing with population behaviors on the bacterial level, and one of the reasons we study them, it's bringing the effects of those genes and proteins much closer to each individual organism's actual actions than we can get to if we were doing that in humans, right? So in other words, you are not just your genes. We as humans are influenced by other items, other aspects. which bacteria don't have that. Okay, so with all of that though, I think that there are striking themes that are consistent between what I have read and understand from my colleagues in animal and plant and other eukaryotic ecologies, and that which we see in the microbial world. And I think that what that says is that there are likely aspects of group dynamics and collectivity that are selected for over time, and those processes continue to be selected for. And so here's what I mean by that. One of the things that we see that's consistent is that for Proteus mirabilis, at least, we do not see a role for the identity-based recognition under situations where there are rampant resources. In other words, if they are in liquid culture or spaces where they can be individual and they're moving around, they can get around far from each other, the IDS genes are barely on. And even when they are on, we don't see any of this separation or changing or things like that. In fact, we don't even see the type 6 machinery form their long swords necessary for stabbing each other under liquid or minimal meter, under liquid or high conditions. We only see those swords form when they're on a surface and they're entering into this elongating stage. And when they're in that elongated stage, they have to move against each other. Their flagella actually move with one another. So it's only during periods of collectivity that we see the swords come out and we see identity play. This is true also for dictyostelium. Dictyostelium has self versus non-self recognition. And when scientists have seen those genes really produced is during periods of stress where the cells have to come together to form, individual cells come together. So I'm sorry, dictyostelium is a slime mold that's usually individual single cells. They come together to form a larger a larger body containing multiple cells of dictyostelium called a slug. And that slug is able to move 10 to 100-fold larger distance than any individual dictyostelium cell can. And it's on the formation of that slug and its migration that dictyostelium makes its recognition factors. And so when these cells, they come together when they need to do a behavior that is much bigger than any cell can do by itself, Proteus, dictyostelium. But it's also during those periods, there are periods of stress. It's a period where the reason they're forming these colonies is because they're trying to get out of Dodge. And so during periods of stress and they're getting out of Dodge, they both have to work with somebody who's different. But then when they get to what they want to separate and keep it for their own kin. And I think as a human, I think that there's a lesson to be learned from that maybe, sometimes when resources are limited, or we feel that resources are limited, or we feel that we need to be protective for those that we might feel more kinship from, maybe that as a human is exactly the time when we need to not do that. When we need to rise above and try and reduce our, basal instincts, let's call it that way. So that's just one area. Another area, by the way, that where I see it is that there's a lot of distinction in a bacterial colony. We like to think of a bacteria as clonal, but for anyone who's looked at gene expression or protein expression, they're not clonal. There's a huge amount of diversity in gene expression and protein expression in any given bacterial population. And with Proteus, it's great. You can even see it with their cell sizes. And so diversity is part of what makes the community interesting, and that diversity is necessary for the community to survive. And so I'll leave it, I'll let everyone take for themselves what they want from that.
Follow-up question to that. Do you see the proliferation of the type 6 secretion system when they're surrounded by like large amounts of non-self bacteria from like different taxa? So if they were to actually be like in the microbiome of a gut or something.
Exactly. So what we have seen with Proteus is that upregulation of the type 6 secretion system actually is it's actually through the self pathway, right? So they don't upregulate it when they're around non-self. They upregulate type 6 when they're around self, which is kind of counterintuitive. And I think that the reason is because they don't really use the type 6. So when they're in your gut, they're in that, kind of quiet, they're in this quiet stage. They seem, we find it in the guts of humans and dogs, etc., but they're not causing disease. And it seems as if they're actually have found a niche. And when they're found with corals, they're protective. They're part of the protective microbiome, reportedly, in coral. When they're moving, so when they form that type 6 is when they're moving. That's when we see them upregulated and turn it on, is when they're moving and swarming collectively together. And when we mix them, at least in my group's hands, when we mix them with Pseudomonas and E. coli and all these other bacteria, it's as if those bacteria aren't there. They literally just swarm around them, just keep doing their own thing. So we see we have not seen a change in the type 6 due to the presence of other organisms. We do see a difference in the presence of self. That, of course, might be different for other bacteria.
That's really fascinating. So we did have another question that came in. How do you see the mechanical influences on the non-adherent signal-mediated contact-dependent bacterial communication?
Yeah, so that is a great, there's so many great questions, and so I'll try to start saying great, but that's another intriguing question. So one of the things that my group has really struggled with, and we still struggle with, is what are the mechanics at all of migration of these large swarmer bodies on the surface, right? Because they're able to move on things as loose as about 0.8% agar. So that's agar that's kind of goopy, but still has solidity to it, all the way up to glass. And so you can imagine that there's all these different forces, depending on whether you're in glass versus a more fluid mechanism. There's actually surface tension on your membrane itself. So it's causing your membrane to kind of do different things, whether it becomes more rigid or more loose. And it's also then force on your flagella. And the part I didn't tell you about is that the recognition signals, they're actually partially regulated by the genes that regulate your flagella. And so they are influenced by the tension that your flagella, they're likely influenced by the tension that your flagella feel. And so we think that there is and then the last thing I'll say is that at low enough agar concentrations, we can basically bypass the recognition effects simply by changing the rigidity of the media, which was published by one of my students years ago. And so that suggests that there is a role for it. What that is, we don't know. How to tackle it, that is not at all my wheelhouse. And so we are, always looking for, and especially if there's anyone in the audience, for colleagues in either engineering or chemistry or bioengineering who are interested in tackling these kind of questions.
Let's move along. So this is the follow-up of the previous question. So if you have two populations and all is equal between them, except population A recognizes things from A, population B recognizes those only from B, both groups would then recognize themselves as self. So if they are in equal density, virulence factors, et cetera, again, everything's equal between them, and they're sensing presence of self and non-self equally, then how would those population dynamics work?
Okay, we haven't done that experiment, even though I realize we should have. And sorry, and by doing that experiment, I mean looking at it on a single cell level over time, tracking each individual population. We've looked at endpoints and we looked at beginning points. But I think when you use the word dynamics, you mean over the course of time. I hope, Mr. Sylvie, or Dr. Sylvie, I hope that that's what you're talking about. So the dynamics over time, that's exactly where we'd like to go, is looking at not just what's happening inside the cell, but as those cells that are in equal ratio, when they start, over time, how does a cell the question we're directly asking is, where is the cell going? Is it changing its migration rate? Is it changing the number of times it interacts with other partners? Is it changing its direction of migration? And then of course, is it changing gene expression? So in terms of the dynamics, we don't have those answers on the single cell level. As a population at the endpoint, what we again see is that one population will dominate the swarm. So one population will be on that, especially if you let them go those 3, 4, 5 swarm circles out. So we can use bigger, we have giant plates, so you can let them go for a long time. Eventually, one population wins out, especially in the situation where you have the rest of the context, the rest of the genome. The exceptions are as follows. If we remove the type 6 in both of those populations, we actually see them stay at about a 50-50 ratio. So that tells us that most of what's happening is actually based on the type 6 secretion related genes, and those can differ between protease isolates. The other place where it differs is that we can actually cause them to never go through a stopping phase. So we can either through genetics or through altering the media conditions, cause cells to always stay elongated and never go through that stop stage where they have to divide. And in that situation, everything just mixes together, moving outwards. So only if they stop and make the concentric circles do we see this population separation or the geography of it. If you don't stop, don't know anything's any different. So I think that's also something just to keep in mind as you go away, is that, you know, these cells, if you don't receive a message, if you receive a message you don't understand, but you just don't stop, great, you can stay part of the population for better or for worse.
One other thing that I was wondering about is when you started this off, you were talking about like your Twitter presence. And I was curious because your work is potentially like somewhat foreign, I guess, to like the general population. When people start talking about microbiology and especially like very specific factors or proteins, the general population that doesn't study those things can get a bit lost. So I was wondering if you, I guess, have any hints for people that work in a similar field as to how they could better communicate their science to people outside of their field.
Thank you for that question. So the first thing I'll say is that I have a Twitter presence, but I'm on Twitter hiatus. So I did not put up any signs saying it. My own personal, I needed a break from Twitter. Let's put it that way. Sometimes one needs to focus more on what's happening physically present as opposed to theoretically out there. So for example, today you saw that my talk has a lot of proteins, and of course I don't expect anyone to care that much about each of these identity proteins. I love talking about the proteins because that's what we work on. We care deeply, and the reason I focus on them and give them names and try and give them analogies is that at the end of the day, what unites viruses, bacteria, archaea, humans, animals, is that we all are encoded by a set of nucleic acids, whether it's DNA or RNA. That is something that all of life on Earth shares in common. We all have genes. And so as much as we want to be abstract up here, this is something that unites the Earth, the planet. When I'm talking to audiences, it depends. I would encourage people to shift their message to the audience, to who's going to be in the audience. And so clearly a talk like this, which is to this wonderful Virginia Tech community, many of whom are biologists and other scientists, it's going to be a more detailed talk where I can show primary data. Hopefully you guys don't mind seeing primary data, where I can show primary data, where I can and really dive into it. And while a talk to say, you know, I used to do talks when I was in Brookline to my children's school group, was a very different talk. There's no proteins there. We talk only about the cells interacting, things like that. I actually write my talks out. I still do, and I practice them. And so I encourage people to do that. And in terms of Twitter, you know, as my husband says, he's a computer scientist, he says that nothing is ever hidden online. Anything you put out there, you should know will always be able to be found, no matter how much you want to scrub it. And so I would just say, be very mindful. Think about what is the message that you are comfortable sharing. There's no need to share all of yourself. Sometimes people feel like they have to put it all out there. No, you don't. You don't owe that to anyone. all you owe is, actually you don't owe anyone anything. What I encourage is that if you have expertise, if you are trained in thinking about complex problems, simply sharing your thought process can help. Simply sharing how you got to where you are now will help. And I think sharing some vulnerabilities also helps. I know that it helped me when I was younger, and I would hope that it provides at least a little bit of insights that there are many ways to achieve success in science.
Wonderful answer. And I believe that is all the time that we will have today for the Q&A session. So that'll be the end of that. The next thing will be a meeting with the graduate students. So I do want to thank you on behalf of the Diversity Committee again, and the overall Virginia Tech audience for the talk. It was really nice. It was great to talk to you and hear about your interesting research. And I'll be seeing you again shortly.
Well, I want to say thank you, Karine, right? Say your name one more time for me. Karine. Thank you, Karine and Amber for, and of course, Dr. Moore for moderating this question and answer session and for inviting me. This is really, it's been fun so far, and I'm very much looking forward to my conversations later today.